Hello class, this is Dr. Anderson and we are starting our cellular physiology series. I have divided this topic up into 5 presentations and we'll start with an introduction to the cell and basics about structure and function. So here are our learning objectives. I'd like you to describe the endomembrane system of the cell, so that's the endoplasmic reticulum, the Golgi apparatus, and vesicles, and use those structures to talk about how you go from a gene in the nucleus to synthesizing some kind of glycoprotein secretion and all the things that would happen. Genes are pieces of DNA that code for a protein. So we want to touch on what are the consequences of changes in the nucleotide sequence, how that results in disease, why some diseases are autosomal recessive and others are autosomal dominant. A mutation that we will talk about are mutations in heat shock proteins, which are a type of chaperone molecule, so what those do and why a cell would make a chaperone and what happens when there are mutations in chaperones. Cells build material, but they also have to take it apart, and what happens if they can't do that, and sort of end up with what are storage disorders. So physiologists talk about cells because cells are the center of everything we do. We've had a section on homeostasis, that homeostasis is the process of keeping the environment that the cell lives in stable. Cells are the unit of life. Living organisms are made out of cells. New cells come from old cells. And in order for an organism to have health, the cells have to be functioning and functioning in a coordinated way. So here's a picture of a cell from your textbook. This is called a composite cell, which means it's not really a true cell. The artist has taken every organelle they possibly can and put it into the cell. So of course here's the nucleus. We're going to talk a little bit about gene expression. We're going to talk about these mitochondria. We're going to talk about the cytoskeleton in a subsequent presentation, but we're going to start out with the endomembrane system, and this is what I mean. We have rough endoplasmic reticulum, there's some smooth endoplasmic reticulum, there's some Golgi apparatus, they're sending vesicles back and forth in the production of secretory products. So we're going to talk about a number of things. Gene expression, which is basically making a protein out of a gene. What happens with mutations, that you make a protein but then you have to fold it up, you have to stabilize it, you have to prevent it from falling apart and forming abnormal aggregations. That's where the chaperones come in. Making a lipid secretory product as opposed to a protein secretory product, and then releasing those products. So here's a picture from a textbook, textbook called Copstead. So here's the nucleus. It's surrounded by a double membrane, and in the double membrane there are pores. They're quite elaborate, but things have to be able to get in and out of the nucleus. As you've learned in previous courses, we make a copy of a gene called messenger RNA. Messenger RNA has to be able to get out through this pore to find the external components. There are also regulatory proteins that are made. Those have to make their way back into the nucleus so that they can control the expression of certain parts of the DNA. So we have the nucleus, and it's a double membrane. It's then surrounded by many folds of endoplasmic reticulum. In this case, it's we see here ribosomes are on the endoplasmic reticulum. That means the ribosomes are helping those areas of membrane to make and store protein, and we have other endoplasmic reticulum without ribosomes. That's the smooth ER. We have more in the way of lipid synthesis going on here, and then we send these vesicles to the Golgi. The Golgi does then some post-translational modifications, adds various kinds of sugars, puts a lipid and a protein together, cleaves things, does some additional processing, then we get it packaged in a secretory vesicle that has to be labeled in some way. Does the secretory vesicle stay in the cell, or does it get merged with the plasma membrane? So a gene is a sequence of DNA that codes for a protein. So the gene is located in the nucleus, and that piece of DNA is spooled up on histones, or little spool-like proteins. They have to be unrolled and unfolded so that RNA polymerase can get access to a part on the sort of the opening part of the gene called the promoter. So the first part is called transcription. A transcription is basically a copy. So if I record my lecture and then you copy me word for word, something I hope that you're actually not doing, I hope you're just listening, but you would be making a transcript of my lecture. So we're still in the English language. One was spoken and one was written. So transcription, you're still in the language of nucleotides. The RNA then goes out into the cytoplasm and meets a ribosome and gets translated from the language of nucleotides to the language of proteins. So you could take your transcript of my lecture and then translate it into Chinese for your friend who lives in Shanghai that wants to hear about physiology. So you're going to a different language in terms of molecules. Now we're not done yet. You still have to fold up that protein and modify it in order for that protein to work. So when you have a mistake or a change in the gene sequence, remember, if you've learned from your previous classes, that there's something called a codon. There are 3 base pairs that get translated by a transfer RNA and an amino acid. So the sequence of those groups of 3 nucleotides determines what amino acid gets put in there. So if there's a mistake in the gene sequence, you could put in a different amino acid. Now, some mutations are silent. Why is that? Well, there's a lot of redundancy in the genetic code. There's more than one codon that codes for the amino acid alanine. So maybe you get a change, but you still put in an alanine. That protein isn't changed and the mutation is silent. Or maybe you put in a protein that's maybe you put in a glycine instead of an alanine, and it's just not that big of a deal in terms of the change in the protein. So the protein still works the same. The mutation is silent. But if there's a change in the protein such that the protein doesn't work as much anymore, then you can have a mutation that results in disease. So for example, there's a condition, spinal muscular atrophy. It's an autosomal recessive, meaning you have to have 2 copies of that gene. So there's a mutation in a protein in the nervous system, and children then lose the ability to walk and that sort of thing. It's actually a fairly common gene. You don't see the disease that often because you need 2 of those alleles to come together, but if you were to sample the population, you'd actually find a fair number of these alleles in the population. Mutations are of differing severity. Maybe there's a point mutation in that only 1 amino acid is changed. Now that can cause quite a big deal. Sickle cell anemia is a point mutation. There's 1 change in an amino acid, but you go from a normal hemoglobin to a hemoglobin that makes a cell to sickle. Sometimes there can be very, very severe mutations in which a nucleotide is added or a nucleotide is taken away. And that means all those groups of 3 get shifted, so that instead of just changing 1 point on the protein, you've changed the whole bloody protein. And what can happen is the stop codon can show up faster than you would like, and the protein gets truncated, and then it's just, it can't function at all. So this gene is transcribed, and these ribosomes, either free in the either free in the cytoplasm or associated with a rough ER. That piece of messenger RNA then feeds through the ribosome, and transfer RNA brings in the little amino acids. The ribosome then makes the peptide bonds, and in this situation the protein would be fed right into the lumen of this rough ER. There could be synthesis of lipids like steroids and fatty acids and those kinds of things in the smooth ER. Then these vesicles pinching off of this part of the endomembrane system could then be sent to storage or sent to the Golgi. So here's a picture telling us about how steroids are synthesized. So steroids are derivatives of cholesterol. So cholesterol is carried around the body on a lipoprotein called LDL that gets taken up into the cell, and there are enzymes in the mitochondria and the smooth ER that work together to make the steroid. Now steroids would not be stored in a vesicle because they're lipid soluble. They wouldn't stay in the vesicle, but then they can be exported from the cell, and next semester when we talk about the endocrine system, we'll talk about how steroids are actually stored out in the plasma, bound to a protein carrier. So this is a beautiful picture showing vesicles melting into the particular side of the Golgi, and in the Golgi all kinds of changes are made. A protein might be cleaved, proteins might be put together, proteins might be glycosylated, meaning carbohydrate groups are put on them. They might be put together with a lipid to make a lipoprotein. Lipids could be glycosylated, and now you have a glycolipid. And then the job of the Golgi is to package them in vesicles, and those vesicles are labeled so that the cell knows what to do with it. Do I destroy this? Do I store it? Do I make a lysosome out of it? Do I get it ready to fuse with the membrane and be secreted? And that labeling system is still trying to be understood. Here's another beautiful picture of protein synthesis. So we have our little ribosomes that are translating a piece of messenger RNA, and now we have the protein inside the rough ER. It's packaged into a transport vesicle. It merges with the Golgi apparatus, and then it gets processed and labeled. So it's been processed, now this is a lysosome. It's been processed, and now this hangs out as a storage vesicle. Or it's processed to be a secretory vesicle, and then this one would be merged. This would add membrane to the plasma, or maybe there's a sodium potassium ADPase that's being put into the membrane so it can function, that kind of thing. So once big proteins are made, they have to be folded so that you can have final expression. So I want to tell you about a classical study where scientists took some control mice and some mice that were exposed to a treatment. They were put into an incubator and preheated, like putting the little mice in a little mouse sauna. And then both groups of mice were exposed to a pneumonia bacterium, which was pretty lethal. And what the experimenters found is that the mice that had been preheated were better at surviving. Much less of them died. They exhibited less inflammation, and the investigators could see that they expressed a whole bunch of proteins. And because heat had stimulated the expression of these proteins, and these proteins seemed to be helping the mice survive, they were called heat shock proteins. So heat shock proteins was the first name, and now we realize that the category is much, much larger, and so they're called molecular chaperones. So heat shock protein is abbreviated HSP. So if you had a chaperone that was 90 molecular weight, the name of that protein would be HSP90. And it turns out it's not just heat that stimulates these proteins. It can be free radicals, it can be um, exposure to radiation, exposure to chemotherapy agents, all kinds of things. So stress will tell cells to make these proteins, and these proteins have all kinds of functions, but the one I want you to remember is that chaperones help very large complex proteins fold into the appropriate orientation. So a gazillion years ago when I was in graduate school, we didn't know about chaperones. We thought that proteins folded up all by themselves, and small proteins can certainly do that, but your bigger, more complex proteins really need the help of a chaperone to fold in the right orientation. And with stress, proteins can start to denature. Well, chaperones can refold them and get them back into a functional shape. If they don't, then you have a bunch of half-folded proteins that start to clump together, and they form aggregates. So you already know about protein aggregates, whether you know it or not. If you know about Alzheimer's disease, you know that there are amyloid plaques. That's an abnormal protein aggregation. So the chaperone process is actually not working there. In Parkinson's disease, there can be an aggregation called a Lewy body. So the problem is when you get these big clumps of dysfunctional protein, that's an additional stress on a cell that can kill it. So chaperones can keep a cell from dying by keeping its proteins folded and functional. So here are a whole bunch of websites, and you can go look at them if you like. So McKusick-Kaufman syndrome. It's an autosomal recessive syndrome where there's a chaperone mutation. Bardet-Biedl syndrome is another chaperone mutation. Here's an ataxia condition where the mutation is a protein in the nervous system. Here's a myopathy. So a muscle disease where desmin, which is an intermediate fiber, intermediate filament, is aggregating. Congenital cataracts is a problem of aggregation of alpha crystalline protein because because of a problem with a chaperone. So these are rare diseases, but these are diseases that have been identified where there's a mutation in a chaperone. Then the cells are vulnerable to dysfunction and death because their chaperones don't work. So let's put some things together. Let's put proteins and genetics and some things together. So McKusick-Kaufman syndrome is autosomal recessive. Let's stop and think what that means. You need 2 mutated alleles to see the disease. Congenital cataracts are autosomal dominant. You only need 1 allele. So if you were to draw out a family pedigree, let's say there's a family with McKusick-Kaufman and another family with congenital cataracts, which family would have more occurrence of the disease? So this is something that I hope we can discuss in class, and I would like you to think about it and write some answers down on your own, and then we can continue this conversation in class. So we've talked about vesicles sending material to the Golgi to get processed. Vesicles are very important means of transfer. That's how we get substances into a cell, substances out of a cell. So for example, let's look at receptor-mediated endocytosis. So in the membrane is a receptor for a particular ligand. And when a substance binds to that ligand, so our immune cells have a kind of receptor called a toll receptor, and that encourages them to swallow a piece of foreign material and process it for the immune system. That's just one example. Low density lipoproteins carrying cholesterol. bind to a particular receptor and get taken into the cell. So the receptors are in the neighborhood of an inner protein called clathrin. So this is a clathrin-coated pit. The binding of the ligand to the receptor sets off a process where a vesicle is taken into the cell, clathrin and all. The clathrin is then stripped off, and now you just have an endosome or a vesicle inside the cell. This vesicle may fuse with other vesicles. That vesicle may have some kind of labeling, say a ubiquitin, and that would tell the cell that this is something that needs to be destroyed. And you could merge that with a lysosome, and then the lysosome has enzymes in it that would break down that product. Then you could turn around and throw the byproducts out, and so that could go out in the waste. So vesicles and lysosomes are an important way to get rid of things we don't want. Here's some fun pictures showing a vesicle process. So there's some things being surrounded by a vesicle. So here's it looks like our little clathrin pit, and substances bind. They're taken into the cell, and now we have a vesicle inside the cell. So, oh, here's my example. So LDL are in a clathrin pit. Then they're taken into the cell, and then the cholesterol can be stripped out, and then this is a secretory cell that's going to use the free cholesterol to make estrogen or cortisol or some steroid hormone. And here's another picture from your textbook with the clathrin pit merging with the endosome, getting the label to tell it to go to the lysosome so it can be hydrolyzed. Now, sometimes inside of the cell we have a big, gigantic protein that's not in the vesicle. So a very kind of vigorous vein of new research is what's going on with this big structure called a proteasome. A proteasome is basically this little breakdown machine. It's a very large structure, and a protein kind of feeds in here and gets broken down. So a protein gets labeled with a ubiquitin. Ubiquitin feeds it to the proteasome. The proteasome takes off the ubiquitin and then breaks the thing down. So for example, skeletal muscle is full of proteins. Well, those proteins get turned over. So after a few months, all the proteins in a muscle cell, they've all been totally replaced by new proteins. So when proteins start to get worn out, you need to be able to make new proteins and replace them. Well, how are you going to get rid of the old stuff? You're going to shuttle it through the proteasome. So degradation is as important to building. So what would happen if you had cellular waste and you couldn't break it down via the lyosome or the proteasome? Now, proteasomes are still being studied, but there are some heart diseases, some myopathies where they're worried that the proteasomes aren't acting appropriately. We also have some things called storage diseases. So Gaucher's disease, Tay-Sachs, glycogen storing disease. So Tay-Sachs is one we can think about. This is an autosomal recessive condition, meaning you need two alleles. So if you have heterozygous individuals, they're not affected. But if you had two heterozygous individuals who came together and partnered and then had a child, there's a 25% chance that any given pregnancy that they would have a child with Tay-Sachs. This is a particular allele that's in high concentrations in the Jewish population. So couples can go to a genetic counselor, and depending on your heritage, you get an African American couple, they might counsel you about sickle cell anemia. You had a Northern European couple, they might counsel you about spinal atrophy or the chloride channel. shoot, the mucus in the thing. I can't think of the name of this disease. Cystic fibrosis. Wow, there it is, cystic fibrosis. Or if you're of Jewish heritage, they would counsel you about Tay-Sachs. So this brings up the idea of a carrier, that families can have unaffected individuals that are carriers. So what does this say about the activity in Tay-Sachs? The particular enzyme involved is called a hexaminidase. So you know, what would that say about the activity of that particular enzyme?
Love hearing you lecture.
So, I want to thank you very much for listening to this presentation. Think about your questions. Think about the things that aren't making sense. You want to write those down and bring them to class or post them on the forum. We have some things to discuss in class about this presentation. My objective is not to simply re-give this presentation in class, but to discuss certain aspects of it and have activities in class where you apply the information. So thank you so much for studying hard and for viewing this presentation, and I look forward to seeing you in class. Bye for now.